Welcome to the Needham Healthcare Conference. I'd like to introduce Mike Weiss of TG Therapeutics.
Great. Thank you very much. Impressive how many people were able to hang around to late in the afternoon here at Needham. Got to thank Simon for the spot. As I get started here, let me just remind everyone that I will be making some forward-looking statements. I do encourage folks who are interested in TG to take a look at our public disclosure documents available on the line. Just background, our goal at TG is to develop the best possible treatment options for people with B-cell diseases. So in cancer, that's things like chronic lymphocytic leukemia, marginal zone lymphoma, which are two leads. We've got follicular and also diffuse large B-cell. In autoimmune disease, it's multiple sclerosis, where we have a large phase 3 program, and then RA and lupus are things that in the future we think we can also tackle for patients. Ideally, we're going to be developing curative combination treatments, so we don't believe that we can get this done with one agent. We do believe that history has proven that if you can put a number of mechanisms together, you can get closer and closer to curative treatments. So we are focused on developing solutions for the patient, which is ideally a cure versus developing individual drugs. And to do that, you need a pipeline of compounds. And these are all compounds focused on B-cell diseases, B-cell cancers. So our lead compounds, umbralisib and ublituximab, are in phase 3, and you can see the logo, the U2 logo. We also use them together as a combination. We have a phase 3 program specifically for the combination of those two drugs. And then we've built a pipeline of B-cell focused agents, all of which can be used in combination with each other and with U2. I'm going to touch on that a little bit today, but anyone who does have an interest, we can always go back over the pipeline at a later date, but I will cover it a little bit. But for today, I'm going to focus on our two lead compounds and when they're used together as U2. So we have ublituximab, which is our next generation anti-CD20 monoclonal antibody. This has been glycoengineered for enhanced potency. So basically it's a fancy word for we took some sugar molecules off the antibody and that makes it a lot more potent. It enhances what's called antibody-dependent cellular cytotoxicity. And when we talk about enhanced potency, we're referring to enhanced potency over the first generation CD20, rituximab. And this drug has shown activity in patients who are refractory to Rituxan. And we also have an advantage here in terms of giving this drug in shorter infusions. On the cancer side, we're able to give this drug in a 90-minute infusion versus the competitors, which are about 3 or 4 hours. In MS, we're able to give that infusion in 60 minutes versus, again, the competitors who are 3 to 4 hours. So a nice convenience advantage for patients using this greater potency antibody. On the other side, we have umbralisib. This is our next generation PI3K delta inhibitor. For those who are familiar with the class, there's a few of them approved today, but they've had some toxicity and tolerability issues. We think we've overcome those first generation toxicities to a large extent, and this drug has seen activity across non-Hodgkin's lymphoma and CLL. I'll show you some of that data. And then the compound is a once-a-daily drug versus the ones that are approved today. 2 of them are twice a day, and 1 is an IV in the same class. So a nice, again, convenience advantage of this next generation. So why do we think we've, in large part, overcome the toxicity tolerability challenges of first-generation compounds? So the first is it's about selectivity to the target in which you're trying to address. So PI3K delta is a lipid kinase, and this is a screen, a very common screen done by a company that most people use to do these kinds of screens. And when you see a red dot, it means that your drug is hitting that target, and the larger the red dot means the more potent it is to that target. Okay? So the target that all these drugs want to target is PI3K delta. The problem is that the only one that actually is targeting PI3K delta in this screen only is umbralisib. Both idelalisib and duvelisib are essentially pan inhibitors. So they hit not only PI3K delta, they hit PI3K gamma. Those are the two primary hits, and then some of the smaller hits are probably on things like alpha and beta. So we think that part of the toxicity profile, tolerability profile of those drugs is associated with the co-inhibition of PI3K delta and gamma together. We think it's causing this autoimmune-related toxicity. Now, again, we would have less of this. Now, what I could tell you also is, if we go to higher concentrations, at some point we will also engage some of these other. But again, at the doses we're using, we feel very good that we're keeping it primarily to PI3K delta. Additionally, something that the other drugs don't do is they don't inhibit a protein called CK1 epsilon. CK1 epsilon is associated with Tregs, and the ability to inhibit CK1 epsilon is protective of Tregs. So when you go and look at what happens when you block both delta and gamma on the PI3K side, you're going to see the downregulation of Tregs. Here you see the protection of Tregs. So embedded within this agent is possibly its own protection against the toxicity that's associated with some of the other compounds. So in addition to a very interesting story on the science of why we think it's better, you can see that the drug has activity. So this is using our drug alone. This is a phase 1 trial. So this is the early studies that we've done. And what you can see in the areas that we're most interested, marginal zone lymphoma, which I'm going to spend a lot of time talking about today, follicular lymphoma, and then CLL, of course, you can see that the majority of the patients, certainly almost all the patients in CLL will respond. I think it's actually 85% in this study. And about half the patients in follicular and about the same in marginal zone will respond to this drug as a single agent. So very nice activity. This was published in the Lancet Oncology, which is a very prestigious journal. So again, hard to say exactly what the exact percentage response rate each one, because these are small numbers, but it's an active compound. There's no doubt that this is active. So what have we done over the years? We started developing these compounds in 2012. We're now in 2019. What have we done? So we've been quite busy, and we've been broad in our discovery and what we're trying to accomplish. So we have our Unity NHL program. This is single agent umbralisib, and we announced earlier that we met the endpoint for marginal zone lymphoma. So like I said, I'm going to spend a lot of time talking about this, and we have some additional data coming up in other cohorts, and this study is fully enrolled. Then we have our Unity CLL trial. This is, again, the combination of our two drugs together. So we enrolled almost 600 patients here. So like I said, we've been pretty busy over the years, and this study should read out by the end of this year or into early next year. And then the last program, which is by and large the largest, is our ultimate MS trial. These are two studies, comes out to close to 1,100 patients enrolled. And this is the CD20, this enhanced, more potent CD20 alone, and we should have data by mid-next year. So lots of activity, lots of data coming out, but what I'd like to do now is talk about what hopefully will be our first entrance into the marketplace. So we have umbralisib for relapsed refractory marginal zone lymphoma. Marginal zone lymphoma, for those who are not familiar with the disease, it's an indolent or slow-growing form of non-Hodgkin's lymphoma. It is not curable, so while you may live with the disease for a number of years, ultimately, if you don't pass something else, you will die of your disease. There's about 7,500 new cases that come in every year. 3,000 patients, we estimate, will be in relapse and needing treatment. We will be approved, assuming we get approval, we'll be approved in patients first relapse and beyond. So this would be the patient population we'd be addressing. There's no fully approved therapies for marginal zone lymphoma. In fact, there's only one treatment that's ever been approved. It received accelerated approval. It's a drug called ibrutinib, and they had about a 46% response rate, and that was recent, so only in 2017. So just recently added, and none of the other agents in our class have been approved for marginal zone lymphoma. We received breakthrough designation in late January for the treatment of relapsed refractory marginal lymphoma, so we're looking forward to working closely with the FDA to move this toward an accelerated approval. So let me talk a little bit about the design of this cohort of patients that we're going to talk about. I'm going to show some data on. So this is, again, patients with marginal zone lymphoma. They were treated with the single agent, just our drug alone, umbralisib. We gave it to them at 800 milligrams once per day, so that's once per day dosing. We treated 69 patients, and again, they all had relapsed refractory marginal lymphoma, and they all had to have prior exposure to another anti-CD20 monoclonal antibody before they got this drug. And so this is the population. We announced in February that we met the primary endpoint. So we know the study is positive. We had a hurdle that we were looking for, which was between 40 and 50%. We exceeded the hurdle rate. But the study was not fully mature, so we're still following patients that are on study, and we'll continue to track that in sometime toward the end of the year. We'll present the full data from the study for all the patients. In the meantime, we will be looking forward to meeting with the FDA, like I said, under BTD, and if all goes well, the goal is to target a filing by the end of the year. So we were lucky enough to get an oral presentation at a conference that's called the American Association for Cancer Research. It's a very important conference. It was held in Atlanta last week or the week before, and we presented interim data. So we have treated 69 patients. That's what I was talking about. So the final data at the end of the year will be on all 69 patients. In this interim analysis, we showed 42 patients for efficacy and all 69 patients for safety. So here are the first 42 patients and basically what happened to them. So basically we had an overall response rate of 52%. Most notable is probably the CR rate at close to 20%, so nice deep responses. And overall, about 88% of the patients had a response or their disease was stabilized when on the drug. And over here you can see this is a very classic waterfall plot. You know, I love these, they're very visual. Each one of these bars represents a patient on the study and what happened to the size of their disease, their lesions. They have disease and it's in lesion or tumor format. And you can see that each one of these patients were down some percentage. Almost all of them, 86%, had some reduction. And you can see the complete responders here at the bottom. So a really nice visual of what's going on in the patients, and the vast majority of them are getting benefit, and a majority of them are actually achieving greater than a 50% reduction, which gets them to be called a responder. Like we said earlier, part of why we're excited about this drug is the safety and tolerability profile. So it's great to have a drug that can induce responses, but you really want patients to stay on the drug, and the only way to stay on the drug is if it's not too toxic for them. So what's really nice to see is that the drug was generally well tolerated. Grade 3-4 adverse events were quite limited. No events of colitis were reported. That is an issue for other compounds in this class. The dropout rate was reasonable and quite low, in fact, for most studies. No deaths occurred on studies, so that was really nice to see that, again, toxicity, you don't want to hurt a patient. And the goal is to help these patients. Unfortunately, in studies, you do have side effects that sometimes could injure the patient. So here we saw no deaths on study, which is great. And grade 3 infections, again, this has been a real problem for the other class members. In fact, one of the drugs in the class had phase 3 studies, I think 5 or 6 of them closed down because of infections. So it's really nice to see that the patients are tolerating and they're not getting any of these or overabundance of these grade 3 infections. So again, overall very well tolerated, very pleased with that profile. And then, this again is for comparison purposes, because everyone asks us, or just to put the data into relative perspective, these are not head-to-head studies. But what I'm showing here is what happened to the patients in this interim analysis versus what you would find in the ibrutinib, the only approved agent, prescribing information. So on an overall response rate basis, both drugs are about 50% overall response. 46% for ibrutinib. We had 52% in this interim. I think, again, I'd like to highlight the difference in complete response rates. So the depth of response that you can get using umbralisib appears, again, this is still not the final data, but appears to be more than you would get with ibrutinib. And then across, the clinical benefit, which is a compilation of complete responses, partial response, and stable disease, 88% versus 83%, again, in the comparable range. Patients with reductions in tumor burden, comparable. Probably the most important piece of data to me when I look at these things is the median duration of exposure, particularly in a class where there's been so much concern about tolerability and be able to stay on the drugs. So what you see here is that the median duration of exposure for ibrutinib, which is by and large a very well-tolerated drug, and in fact it's used a lot because people perceive it as a very well-tolerated drug. The other drugs, not used in marginal but in follicular, their time duration on study on treatment was about 6 months. So this is almost double for ibrutinib. We're at 10 months and going. We didn't find the median duration of exposure yet for this group of 42. So again, it's a compilation of both activity and safety built into one measure. So I like to look at that, and obviously we're super excited about the median duration of exposure as it stands. Duration of response is also very important. You want to be able to give them a response, but you want it to last. So here again, we're out, with 12.5 months of follow-up, we haven't hit the median duration of response. So that's very good, and hopefully that'll be even longer. And here's another one that's also very important, median time to response. So this is about how long it takes you to actually achieve your response. And here again, we get there pretty much for the median at the first scan versus ibrutinib at the second scan. So I think once we're thinking about being on the market and you're a patient and you're thinking about this, which drug is going to give you a faster and potentially deeper response, I think if this all plays out as the final data comes in, I think we have a nice profile to go to the market with. And the other nice part is, again, while I said ibrutinib is a very well-tolerated drug, it's not perfect, but in its imperfections, they're different from our drug. So again, our drug is not perfect either, but it's different. And what's nice for patients is that if you don't tolerate this drug, or you have a cardiac issue, or you have some bleeding risk, you're on blood thinners, or you have hypertension, these are not the things we worry about with our drug. So again, what we would describe as generally not overlapping in toxicity. So if you don't tolerate one drug, there's no reason to believe you won't tolerate the other drug. They have a different profile. And we've tested in patients with CLL, we've taken patients that don't tolerate ibrutinib, and we put them on our drug, and they tolerate it very well. So we've already shown that, at least in CLL, and that'll help us segue, of course, now into probably one of our biggest programs, which is Unity CLL, which is our big trial that I was describing earlier. CLL is a pretty large problem. Here in the U.S., there's about 115,000 Americans living with this disease, 20,000 newly diagnosed patients. We showed earlier that we have very nice activity as a single agent in CLL. This study, this Unity CLL study, is being conducted under special protocol assessment. And what's really unique about this trial design is that if we are successful, and we believe we will be, this combination, both drugs will be approved, and they should be both approved for newly diagnosed patients and relapsed patients. So remember when I was talking about marginal zone, we looked at the incidence of new patients and the relapsed patients, and there we'll be approved in relapse. Here we can be approved in all patients. So there's 20,000 newly diagnosed patients. There's probably almost a similar amount of relapsed patients every year. So it would be a very broad and large label. The hope is to have data from this study by the end of this year or sometime early next year. And like I said, we enrolled 420 patients in these two arms, but in total in the whole study we ended up enrolling 600 patients. So a really nice large database of information for us. So why are we so confident that this is going to be successful? So the endpoint for this trial is progression-free survival. And basically it's a measure of how long the patients are alive and have not progressed from their disease after you've given them your treatment. And I want to point your attention to, first, the green line. This is from our phase 1 experience. This is the progression-free survival for single-agent umbralisib. So this is just one drug by itself, and the median is at about 24 months. That's significantly better than you would expect from the control arm, probably almost double what you might expect to get from a PFS from the control arm. But that's not what we're studying here. We're studying umbralisib plus our CD20, and that's the blue line. And what you can see, it gets relatively flat, and we're out now 20, almost 30 months and ongoing. So, and again, if you would expect the patients in the control arm to be about 12 months, I mean, their median would be reached over here. There's nowhere in sight. So again, in theory, if this plays out in our phase 3 trial, we're going to be wildly positive. So again, we believe that we're going to be successful. We feel very confident from this kind of information that we're going to have a successful outcome. And again, the goal is to have this hopefully done by the end of the year or sometime next year. So CLL, as I mentioned, it's a major disease. Like I said, over 100,000 people living with the disease. It's one of the fastest growing markets in oncology, hematology. The expectation is by 2025, it'll be over $10 billion. So this is a major market opportunity. And so as I transition, I'm going to do a little transition between here and MS. We'll clear our heads as we get into MS. But what I'm going to leave you with is what's next after we've finished Unity CLL, after we've finished Unity NHL, and ideally after all that's done, we're going to be on the market for the treatment of non-Hodgkin's lymphoma, for chronic lymphocytic leukemia. Then the pipeline comes in, right? And like we said, we're trying to create these combinations for best outcomes. So one drug is not good enough. 2 drugs is probably not good enough to consider a patient cured. But once we start adding 3 drugs, 4 drugs, or 5 drugs, perhaps we can unlock the key to this. And we already have some very interesting data using U2 with a similar mechanism to our own pipeline drug of 1701. And what you can see is in a lot of these diseases, 100% response rate, 100% response rate, 100% response rate, 80% response rate. So we are very excited about the ability to put these drugs together in a triple. And likewise, we're very excited about taking U2 with our drug 1501, which is an anti-PD-L1. Again, we've studied a similar compound that's available in the marketplace, and in CLL, 90% response rates. Richter's transformation, a very aggressive, transformed version of CLL, 50% all CRs, very nice activity. And again, we can start to even package not just 3, but 4 and 5 of these together. But again, that's for later. We'll get to that in 2-3 years. That'll be a really big story. In the meantime, the next up is going to be our MS program, which is the biggest of all. So we thought, we have marginal zone, which is, hopefully on a nice linear track toward approval. We're going to get CLL data. We're confident and believe that that's going to be positive. And then as we move into MS, everything just gets even bigger. So this is our CD20 alone. There's about 1 million Americans living with MS. This is, again, a major, major problem. We've already completed our phase 2 trial. We presented the data. I'm going to go through some of that data. And we've already fully enrolled our phase 3 trial, and this is also being conducted under a special protocol assessment. So this large phase 3 trial, again, 500 in each arm. I think we over-enrolled a little bit. I think in total we're closer to 1,100 between the 2 trials. This will read out sometime the middle of next year. So it's getting close. Talk about the data. Why do we think that this study is going to be successful? So the phase 2 data, the primary endpoint for the trial and the primary endpoint for the phase 2 trial is what's called annualized relapse rate. I don't think it's going to make any sense other than to say that it was 0.07 at 48 weeks, which is really low. And just to give you a sense, there's one other drug approved with our mechanism for MS. And at week 24 in their phase 2 trial, they didn't present their week 48 data, which would be comparable to this data. They had a 0.13 annualized relapse rate. Oh, by the way, lower is better, just in case anyone was curious. lower is better. And at a similar time point in our trial, we had 0.05. So again, at least shaping up to be at least as active or maybe perhaps even more active. And the other thing that's really important about this data is to see how well conserved the annualized relapse rate was when they went from 24 weeks and 55 patients to 96 weeks and 1,600 patients. So the scale up between these two trials, you're going to a lot more patients, but yet the annualized relapse rate is very well predictive of what they got. Again, do we feel like this should be very well predictive of what we should get? Yes. And anything in this range is going to be great for us. Everyone loves a pretty picture. The patients who have MS will also come in with lesions. It's one of the telltale symptoms of what's happening. You can scan them and you can see these lesions. Every patient that came into the trial, every single patient that came into this trial with a brain lesion, the brain lesion disappeared while on the trial, and no new lesions were formed while the patients were on this trial. So even if you came in without a lesion, you didn't get one, and if you got rid of them, you didn't get any new ones. So again, very, very successful phase 2 data. In terms of market opportunity, the U.S. is 1 million. There's another, probably 1.3 ex-U.S. Again, the sales are driven U.S. primarily. The only drug approved with our mechanism was approved about 2 years ago. They're on almost a $2.5 billion run rate. They'll do, I think, for their second annual year in sales, they were greater than $2 billion. So a remarkable launch. The data on this class and in this drug look great. They've already captured 12% of the MS market. The expectation is that is growing. So our value proposition in MS, we see equal to better activity with comparable safety. That's our goal when the program is done. We see a convenience advantage. We have a 1-hour infusion every 6 months versus 3 to 4 hours for the only other drug that's approved. And the one nice thing that we have in our back pocket is we can be very strategic about how we price the drug. We're not going to be protecting a very high price and what now is a $2 billion, which may be a $4 billion by the time we get to the market, franchise of the other company. We can be very aggressive if we want to be on price. And we see this as a real opportunity for us into the billions in sales for ublituximab in MS alone, on top of what we think we'll do in CLL and marginal zone and potentially follicular and others. So we've had a great first half of the year in terms of achieving our goals. We pretty much knocked off everything we were seeking to achieve pretty much by the end of the first quarter. Second half of the year, hopefully things are going to really continue to accelerate. Again, hopefully we'll get the events required for our Unity CLL PFS trial. Hopefully we'll be able to file our NDA for our marginal zone cohort from Unity NHL. And then we should have some really nice pipeline updates and Unity NHL updates later in the year, hopefully at ASH. So with that, I'll leave you with some corporate and financial information, and I thank everyone for their time and attention.